Welcome to Chapter 13. Chapters 13 and 14 cover the basics of cellular respiration and photosynthesis. These two concepts are broken down into two parts. In Chapter 13, we are going to focus on the first part of cellular respiration, and in 14 we will talk about the process of oxidative phosphorylation using our good friends the membranes. Here are our objectives for the chapter. Make sure you know these by the exam. If you have any questions or need help on a topic, please let me know. As I mentioned, for Chapter 13, we are going to depart from our membrane talk, and briefly, until we come back to oxidative phosphorylation in Chapter 14. In this chapter, we are going to mainly talk about how cells catabolize sugars and fats using glycolysis in the citric acid cycle. We are also going to talk about how the cell regulates these processes. So far this semester, we have talked a lot about the processes in the cell that use energy, but we haven't talked about how cells actually get this energy. In this section of the lecture, we are going to talk about how the body and cells break down the large food molecules. As we know from learning about glycolysis and cellular respiration in other classes, sugar is an important fuel for the cell. We also learned in previous courses that energy is obtained from the oxidation of the sugar molecules through a three-step process, including glycolysis, the citric acid cycle, and the electron transport chain, coupled with ATP synthase. If you remember from Chapter 3, the energy required to break glucose down to CO2 and water in one single step would release an exceedingly large amount of energy and have a huge activation energy barrier. Instead, we break the sugars down step by step to make sure that we can extract as much energy as possible from the cell without causing damage to the cell. But we're getting ahead of ourselves. Let's first talk about how we get glucose and other macromolecules from the food we eat into cells. The entire process of breaking down our food molecules is called catabolism. The first step in catabolism is digestion of our food into smaller molecules, usually before they enter the target cell. Hopefully you are aware that this occurs in our digestive tract and is accomplished through specialized enzymes. Sometimes larger molecules can be digested in the cell inside an organelle known as the lysosome. We will discuss the lysosome in more details in Chapter 15. Once the food is broken down into smaller molecules, it can be transported into the cell via one of the transporters we talked about in Chapter 12, for example, the sodium glucose pump. Once inside the cell, catabolic processes you have learned about before start. Let's take a look at these processes, starting with glycolysis. Glycolysis is the second stage in breaking down sugars into usable energy. As you can guess from the name, this step is all about splitting sugars, hence the glyco in glycolysis. This stage occurs completely in the cytosol of the cell and occurs in all cells, including prokaryotes. In fact, the presence of glycolysis in all living organisms is another one of the arguments in the theory of evolution, that all cellular organisms descended from an original ancestor cell that had this pathway. Glycolysis is the only step of the three involved in cellular respiration that can occur entirely without oxygen present. In simplified terms, the process of glycolysis is as follows. 1 molecule of glucose enters, 2 molecules of pyruvate leave with a net gain of 2 ATP, along with the reduction of 2 NAD pluses to NADHs. That's a simplified version, and it may be the only version you are familiar with. Let's take more of an in-depth look at this process. There are 3 parts to glycolysis. They are the energy investment phase, the sugar splitting phase, and the energy generation phase. In total, there are 10 steps in this process. Since you will need to know this stuff in later courses, I expect you to memorize all of the intermediates with their structures and the names of the enzymes. This will help you to understand the purpose of each step in the process. So let's look at each step now. For each of these slides, I'm going to talk you through the importance of each step and the change in molecular structure. A good study technique would be to summarize the importance of each step in your own words so that you can get a good feel for each step in the process while you draw out the change in the molecular structure. The goal for step 1 is to prevent glucose from exiting the cell. What type of molecule do we know of that cannot easily diffuse across the plasma membranes? Anything with a charge. To do this, the cell uses one of two ATPs expended in the investment phase. The hydrolysis of ATP will add a negatively charged phosphate to the glucose molecule, making it a glucose 6-phosphate and giving it a negative charge. This makes sure the glucose will not leave the cytosol. In step 2, we change from a 6-carbon sugar ring to a 5-carbon sugar ring. When this rearrangement happens, the molecule becomes almost a mirror image of itself. See the aldehyde group sticking up from the fructose ring? This makes it more able to be split evenly down the road. This is performed by an isomerase, specifically phosphoglucoisomerase. This gets us one step closer to being ready to split the sugar in half. By the way, the fructose that we consume in fruits, honey, or dare I say it, high fructose corn syrup, can enter glycolysis at this step by having an enzyme add a phosphate to it. In step 3, we finish the preparation for sugar splitting. In this stage, we use the second of two ATPs to balance the molecule by adding a second phosphate to the other side of the molecule. See, we have a phosphate group on the other side to form fructose 1,6-biphosphate. This stage also serves as the gatekeeper to the rest of glycolysis. Any monosaccharide that is going to be broken in glycolysis, yes, and more than glucose can go through this process, has to pass through the enzyme phosphofructokinase. In step 4, we enter into the sugar splitting phase of glycolysis. In step 4, the 6-carbon sugar is split into 2 molecules by the enzyme aldolase. The two products of this splitting are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. In the figure, we have flipped the glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate has to be converted into glyceraldehyde 3-phosphate by triose phosphate isomerase. This is because only glyceraldehyde 3-phosphate can pass on to the remaining steps of glycolysis. So the dihydroxyacetone phosphate has to be converted to glyceraldehyde 3-phosphate. See, and what we do is we take this and we switch it with this group here. From this point on in the process, everything will happen in sets of 2 because there are 2 glyceraldehyde 3-phosphates. In step 6, we transition to the energy generating or harvesting phase. Remember that from here on in, everything that we look at will happen twice. See the little red 2s I've added everywhere? Before we start this energy harvesting phase, let's remind ourselves of what we are trying to harvest. Our goal at the end of this process is to obtain 4 ATPs and 2 NADH. In step 6, we accomplish the first of these goals by harvesting 2 NADHs. This is accomplished by the oxidation of glyceraldehyde 3-phosphate. The NAD plus gains an electron to become NADH. Step 6 is important not only due to the formation of 2 NADH, but also because in this step the glyceraldehyde 3-phosphate gets another phosphate group added to it to become a very high energy anhydride ready for later reactions to occur. In step 7, we form the first of our 4 ATPs. The transfer of the phosphate group occurs from the 3-biphosphoglycerate to ADP via phosphoglycerate kinase, which creates 2 3-phosphoglycerates and 2 ATPs. What we are left with here is a high-energy phosphate group on the number 3 carbon molecule. In this step, the remaining phosphate ester linkage is moved from the number 3 carbon to the number 2 carbon. This creates 2 phosphoglycerates and prepares the molecule to give up the next phosphate. In step 9, we remove water from the molecule. This creates a new double bond in the molecule, and it creates a high-energy enol phosphate linkage. This is the final change before the molecule gives up its phosphate group. Phosphate kinase will remove the phosphate from phosphoenol pyruvate to create pyruvate and ATP. This completes the process of glycolysis. We have gone through the investment phase, the sugar breaking, and the energy generating phase of glycolysis. The net gain for this process is 2 ATP, 2 NADH, and 2 pyruvate molecules. Remember, the main goal of glycolysis is to create NADH for the electron transport chain. The ATP is a good side bonus. The ATPs that are formed during glycolysis are formed by a process known as substrate level phosphorylation. The NADHs are formed through redox reactions. Let's take a look at this in a little more depth by examining the redox reaction of step 6 and the coupled substrate level phosphorylation reaction of step 7. In these reactions, enzymes use coupled reactions to help the cell harvest usable energy. We are converting an aldehyde into a carboxylic acid using a two-step coupled reaction. We start by forming a covalent bond between the glyceraldehyde, or G3P, and the sulfhydryl group of the cysteine side chain of the enzyme glyceraldehyde 3-phosphate dehydrogenase, or G3PDH. After this covalent bond is formed, NAD+ also non-covalently attaches itself to the complex. The oxidation of G3P will then happen when we remove 2 electrons and a hydrogen ion and transfer them from the G3P to the NAD+, forming NADH. The energy released in this reaction is used in 2 ways. The first is the storage of energy and NADH to be used later in oxidative phosphorylation. The other part of the energy goes into converting the bonds that have been formed between the enzyme and G3P into a thioester bond. To release the molecule from the enzyme, a phosphate group is added to the molecule. This then moves us into step 7, where the high-energy phosphate is removed to form ATP. You can see in the picture on the right hand side of the slide how the coupled reaction works to allow the formation of the unfavorable high-energy phosphate bond. Before we move on to acetyl-CoA transformation, let's summarize what we have just talked about and what is important to know about glycolysis. First and foremost, glycolysis requires NAD+ to run. Without it, the process cannot proceed. Glycolysis yields a net of 2 ATP, 2 NADH, and 2 pyruvate molecules. The main purpose of glycolysis is to create the NADH that is used downstream in the electron transport chain and chemiosmosis. I want you to be able to answer basic conceptual questions and application questions about glycolysis. One more quick note before we move on is your textbook includes a brief discussion on fermentation, but for this class I am not too concerned with this. If you'd like a refresher on fermentation, feel free to read through that section in your textbook. Well, that's it for this topic. We will pick up the rest of aerobic respiration in topic number 2.